So good evening everyone, and welcome back to the last lecture of this year. It's also the concluding lecture of the winter term of our lecture series called Tempor Realities. And tonight it is our great pleasure to get insight into the work of another of our own faculty members, Dr. Petra Gruber, who recently took over the chair of the Building Construction Department here at the Institute of Architecture. So reflecting on the notion of temporality within the domain of building construction is at the first glance fairly straightforward, since the act of construction is a process carried out over a period of time, embedded in socio-economic realities, and once concluded or out in the world, buildings have to stand the test of time. Yet, thinking about the unfolding of processes across scales, from micro scale inside matter or building components to building as growing, are the temporalities Petra is investigating in her research work, rooted in domains such as biology, bionics, biomimetics, biomimicry, as well as material and building science. All the mentioned domains foster interdisciplinary collaboration and can be seen as working hand in hand to inform design practices which revolve around aliveness, meaning processes and interactions. So in this milieu, Petra Gruber is a researcher and educator in architecture and biodesign and holds a PhD in biomimetics in architecture from the Vienna University of Technology. She has previously taught at the Ethiopian Institute of Architecture, the Biomimicry Research and Innovation Center at the University of Akron, Hochschule Rhein-Waal in Germany, at the University of Reading in the UK, as well as the Vienna University of Technology. In her office TRANSARCH, Office of Transdisciplinary Research in Architecture, Petra leads research projects in traditional architecture and on spatial and functional aspects of biological structures, of biomimetic innovation in architecture and the built environment. So, if our first lecture was freely recombining fictions across time and space in acts of anachronic radicalism, and the second one presented snapshots resting within the notion of hyperobjects, tonight we will zoom into the world of bio-inspired, bio-based, and living building matter. In order to do so, the title of the lecture tonight is A Living Architecture. Bio what? And without further ado, please help me in welcoming Petra Gruber.
Thank you, Maya, for the introduction. Thanks a lot for having me tonight. I'm happy to be here, and thanks all for coming, in spite of the oncoming holiday tomorrow, so I'm delighted to see a few people in the audience. And Maya has done a great job introducing myself, so I won't repeat. So I'm happy to be here at the Building Construction Department, seeing my colleagues in the audience too. So hopefully we can have a nice evening in the chat after this lecture together. And so my talk today is about bio what. So referring to the topic of Tempor realities, I want to talk a little bit more, as usually, about processes and maybe also give a few hints on timelines, which may be very different working with biological structures than with technology. So I would like to give a bit of an introduction into the methodologies that are around and want to use my research projects to illustrate some of the different approaches. And those projects have very different timelines. So Biornametics was a one-year FWF project funded by the Austrian Science Fund. Growing as Building was 2.5 years. All of them extended a little over time, so the publications took another 1 year, 1.5 years to materialize. And Mycoterials is a research that has followed me since we started the Growing as Building, and I did a lot of stuff in Akron too with PhD students, so it spans a time frame of, I would say, 5 to 7 years. And then the last two projects, Leaf, Shape, Thermodynamics and Root Research, are both more than 5-year projects done by PhD students. So this gives you another scale. So knowing that, you might also see those pictures that I show you a little differently. It's just a very brief intro in what the PhD students and a lot of grad students, undergrad students and myself did together over a long timeline. And in between the projects, I want to talk a little bit about methods and processes and show you some of the workflows that were important for the project. And so jumping in, I would like to start with the most important term at the moment and the most generic term that I found to encompass everything. And so currently I work with this word of biodesign. And I would say that as we have reached a state in our kind of human development where we face so many very, very complex, difficult problems with the current technology, I think it's time to look at another example. And the only other example than human technology that we have is nature and biology. So I think it's time to really have a closer look at what we can do differently and using biology as a paradigm for our new strategies. And I'm very happy to see that since I did my PhD in 2008, this field has exploded into a variety of directions. So bio what is like just a funny, subtitle to convey the notion of that there's a lot out there in definitions and different areas. So this is just to give you a glimpse of an intro into those different fields. So the work that I started with was biomimetic, so taking inspiration from biology, taking over principles into technological design, but since has expanded into really bio-based or biomaterials design, where we actually use living organisms integrated into materials and into architectural design projects, and ending with like living building materials that actually are alive and growing buildings, concepts of having real living buildings that we inhabit. So those definitions come up in different contexts, and it's interesting to see that everybody's working on methodologies as well, so those are a little different. So be aware of those different terminologies as you are inspired and go into the literature. My own framework is biology, as I was really always very interested in biology. I have a really good background. I studied pharmacy, I did a year of botany, so I would say I have an undergrad degree in botany. So I was really annoyed in architecture by this, takeover of terminology, and the purpose of my PhD was to clarify those fields a little bit. So I looked at those signs of life in biology, trying to understand what are we talking about with living systems, and it's not so straightforward. There's many organisms that exhibit one or the other sign of life, like viruses, for example. They are not technically alive, but they are in some way. So it's not that easy to define. If you talk to space researchers and people who are in astrobiology, you will also find that that's really something that we don't really know how do other life shapes exhibit themselves. And so we work still with a classical set of criteria of life that we apply. So we say if something exhibits order, propagation, growth, energy use, reaction, metabolic activity, homeostasis, and evolutionary development, we call it alive. So we have to use all those criteria, and then we say this thing is actually alive. And there's a few more, like information use, self-organization, and limitation. Organisms are limited in space and again time. So this is another notion that is really important in the context of this lecture series. So what I did in my PhD is I took those terminologies from biology and used this as a filter to look at architecture and the built environment. And the result was that it seems to be a valid paradigm in architecture development right now, so that a lot of people are interested in those criteria of life and implementing those criteria of life in architecture and in the design process. And this has profound consequences to architecture. It changes the notion of a very stable, rigid environment to one that is very dynamic, responsive, wet, resilient, self-healing, and so on. All those features that we would like to see in contemporary development. And what that means for different technical categories you can see on this slide, which I don't want to go in further detail because you're all aware of this. But I think still that biology is a concept that brings that all together. And coming back to evolution, I think that the field of traditional or vernacular architecture is one that can serve as another example of like natural construction that has developed over very, very long time frames and encompasses information that is not conveyed in science or in any other way where humans would have access to this information. So the buildings themselves store what was important in former times and probably still is. And one of the best examples is the architecture on Nias, Indonesia, that is exceptionally well adapted to the earthquake situation on that island. And also the location of the villages is just ingeniously adapted to the situation there. And this is something that I did research with a group around the Vienna University of Technology, and it was also a case study for my thesis. I've done a lot of research in different countries. I will not show this today. I hope to bring that into the construction department and have more talks about that in the future. And talking about biology, the most striking feature about biological design for me is this inherent connection between form, function, and structure. And in biomimetics, in this effort to translate from one side to the other, function is always or in many cases, function is establishing the relationship. So what we are doing is we're looking for specific functions in biology that we seem to have problems with in our current world, in technology. And here you have a simple example of a rainforest plant, and you see some very simple features. You see tissues that serve different purposes. So one you can discern is a leaf. So the leaves are responsible for photosynthesis, for energy capture, for metabolic activity. And then you see those tubular structures that are spreading over the space, and they are actually there for spreading and also for transport. So they have another functionality and they look differently. And this is just a very simple example how biology functions. And this happens in all kind of scales. So on the side you see a section through a leaf, and the kind of circular shapes are all cells. And the cells are very differentiated depending on the location on the leaf and the functions they fulfill. Some of them are skin tissue, some of them are energy capture, some of them are like arenchyma for gas exchange. So the differentiation is according to the main functionality. And it's not always easy to capture the main function in biology too. I've talked about that in the Circular Strategies Symposium when I focused on mycelium materials. So this is just the most important feature that I find really interesting. And one of the first projects that we actually did at the Angewandte, that was funded by the Austrian Science Fund and that I co-led together with my dear colleague Barbara Imhof, was called Biornametics, and researched patterns from biology for a potential translation into architecture and design. And it was very interesting to have this happen in the artistic context. So we were not forced to generate a specific product. We could freely venture and navigate this project. And what you see here is the final installation. On the way, we researched I think, hundreds of biological role models, and we documented I think something about 50 of them. And what you see here is the relationship between those what we call role models from biology and a certain function that we were interested in. And as architects, we are always interested in shapes, shape change, color change, things like that. So we had a range of functionalities that helped us also then translate this into a design project by finding a combinatorial game and framework for our design proposals. So that's one of the characteristic elements of biomimetic design, that we look at several examples from biology and then trying to find the best champions, the champion adapters. So examples that really serve a purpose and that are really well adapted for this specific interest that we have. Another example here that shows you also the digital translation of principles is the really simple example of the growth of shells. They exhibit rim growth, so they just grow these shells at the edge, so they just add material in several stages. And what we did is we just took those shells and we tried to mimic this process in rhino and grasshopper with parametric design principles. And then it becomes really interesting when you leave the confinements of biology and just use those growth principles and totally different shape formation that you can then use as an inspiration. So this is a truly biomimetic approach that we have used in this project, and we followed a lot of different design strains, and only some of them then materialized in this big installation. Others were just documented with video, and for others we built models and small simulations and digested all the materials in a video production. So I think the value of this project is also that there is many outputs in different fields, publications, as well as models and so on. And coming back to the biomimetic process, this is the scheme of what we're doing. So we're trying to connect science and design with this information flow, in this case between biology and technology. And the most important piece is the translation process, so the abstraction that happens, moving from one side to the other side, is really important. And as an architect, we cannot do really a lot of basic scientific research in biology, but what we can do is we can take the research result of biologists and use them in our design projects. And another very interesting thing happens on the way. As we are interested as architects and designers, biologists understand suddenly the value of the information that they generated and the specific things that we as designers are interested in, and in this way inspire more basic research. And we call this process reverse biomimetics. So due to the interest to translate, also the life sciences get infected and start researching the topic in a new way. And so this kind of spiral of knowledge is really something that is really cool and interesting and has been the frame of my work. And then we were lucky to get a bigger research project, Growing as Building, and we could explore growth from biology for architectural design for 2.5 years. Again, here at the school, we were attached at the Studio Lin, which was a really great platform, and worked with the students on some of the projects. We also had research assistants, which is always one of the principles of my project, to also integrate teaching into the research. And it was the first project where we had our own biolab. So we actually grew stuff in the architecture lab, and that was as challenging as it was interesting. So we worked with slime mold, algae, mycoterials, and we researched many other role models from biology again. And here we had those growth-related principles as a negotiator between our architectural interest and what we found in biology. So we had, again, this information transfer between architecture and biology via those growth principles that were so cool to research and we were interested in. One example is the slime mold as a biological example, and many of us know it's a unicellular organism that exhibits still as a colony an amazing intelligence in pathfinding. It's very efficient and finds the shortest way to a food source. And we managed to use this capacity in a three-dimensional setting and used the slime mold as a co-designer for a design project. So this is actually already a lot more than biomimetic. So we don't only use principles from biology, so we actually use the knowledge of this organism to do a design project. So this goes beyond biomimetics and is actually bio-co-design or biodesign if you want. So what you see here is the slime mold growing through a 3D printed model of the Mount cell force. And this is the slime mold is defining the design project that we then designed in conventional software. So we scanned the growth of the slime mold and used the connectivity of the organism for the project. So that's how it worked. If you're interested, go and see the paper publications that describe the process in more detail. And while doing those thematic projects, we also try to push technology. In this project we were working with a cable suspended printing technology, trying to free the printer from its cage and trying to make larger scale printing possible, also trying to get a cycle of material going with the algae, printing with sustainable materials and so on. So in the projects, technology is always a topic and we always try to push the boundaries here too. And shifting to a real research university, going to the University of Akron in the US and working on the Biomimicry Design and Innovation Center was shifting my work towards basic science even more. We continued to work with those micromaterials. I just want to show you two slides about this today because I talked about it in the Circular Strategies Symposium quite a bit. So you see here one exhibition piece. This was one of the largest panels that we did, again with the growth of fungi. So this is a composite material that exists out of organic substrate that is actually shapeless. So it's like could be paper chips, wood chips, any kind of organic material that is overgrown with mycelium, with a mycelium network that solidifies the material into something that is really like a lightweight composite. And if you keep the mycelium alive, it looks like this. So the fungus grows into fruiting bodies, spores emerge, and then it gets a little bit tricky for using it in human spaces because spores could affect us, our health. They invade our lungs. They could be toxic if we take the respective species. So what we try to do if we use it as a building material is to kill the fungus and have the material as an inert, dead, bio-based material, as an insulation material for similar purposes. So it makes a big difference if this thing is alive or not. So be aware of this too if you look at those research projects. We did a long-term study. We did a study for about 2 years. We exhibited these materials with different post-growth treatments in the field station of the biology department, where people could actually go there and see what was happening. We monitored the decay, and so depending on the treatment, you could say that it's about a year that those elements remain in nature, in the outdoor setting. After 1.5 years, the first panels start to disintegrate completely, and after 2 years, pretty much all the panels had reached a stage of disintegration. Some of them really had evaporated and were kind of spread into the environment. So that was a really interesting take on it. I think it was one of the first outdoor studies that was done with this material. And so talking about materials, we differentiate between bio-inspired, bio-based, or even living materials, and there's many examples in architecture that you can look at already. So materials research is a big part of this research in general. And in the following, I want to switch to two of the really large studies that we did in Akron. One of them was the leaf shape thermodynamics study that was carried out by my PhD student Ariana Rupp. And she got interested in plant biology and plant leaf shapes in general. Again, we tried to understand this shape-function relationship and to uncover how plants were actually managing the change of the environment. So that's the reason why plants are so interesting for architecture, because they can't run away. So you don't have behavior in the sense that animals exhibit it. They cannot hide. They have to stay at the same place and have strategies to cope with a changing or adverse environment. And those strategies are really cool. So the thermodynamics of leaves were the example that we were interested in, and you see here just a small fraction of leaf shapes that are possible. And I've talked about that previously. Biology is always multifunctional, so thermodynamics is just one of the aspects that defines a leaf shape. There are many others, but it seems that for some of the shape features, thermodynamics are crucial. And one of the features are the edge formations, so the edge geometry of leaves in connection with the overall shape seems to be responsible for the thermal properties of leaves and how they cope with overheating and cooling. And to understand this, Ariana did a lot of shape studies. So she scanned and drew leaves and created parametric models and abstracted the shapes. And what you see here are very simple biomimetic crude models that were made by parametric design. And their models exhibit the same surface area and the same boundary length, just the shape at the edge is different. And what we can clearly see with these very simple evaporation studies is that the shape makes a big difference. So depending on the scale of the system, you can see that for some of the shape, the evaporation is totally speed up. And for us, this was really exciting as a principle, because cooling is one of the big issues in technology and in architecture in general. And those very small shape features could be exploited to a way larger extent to work towards more efficient cooling systems. And so that's what we did. We experimented with this, and Ariana took it on as a PhD study, did a lot of biological and biomimetic groundwork. So we had literature reviews on the topic, we did a lot of geometry definition. Again, this is something like reverse biomimetics, so biologists have never looked at it that way, so there's no proper leaf shape definition that talks about evaporation efficiency. So what we needed to do is to define those geometrical features, and so we came up with the LDI, which is a relationship between the perimeter and the leaf area, and a measurement for the solidity of the leaf, which talks about how fuzzy the leaf shape actually is. So a combination of those geometric features then gave information about the evaporation capacity. So it's all not that straightforward and all like fraught with a lot of reading, a lot of interdisciplinary work, and a lot of experimental biology. So a lot of this research is happening in the realm of biology. And Ariane also had to understand the basic physics of the system that is pretty complicated. So what happens here is the physics of flow. It's a very complex thing that is not easily grasped in mathematical terms, and everything boils down to what is called the boundary layer theory. And simply put, it's this kind of area where the humidity from that soaked leaf transfers to environmental humidity. That's a very small layer, and this layer thickness is scaling the system. So even if we have come across this very cool effect, it comes with the sad precaution, we cannot just scale it up. It's only usable at that scale that we find it. And that's very often the case with biological principles, that they apply to a very small scale, and we can sadly only translate it in materials or surfaces and not in like building scale or building elements. So here you see the lab setup. So we have this infrared camera that gives us information about temperature and talks about where is evaporation happening. Then you see a digital scale that gives us information about how much water is already lost. So we can exactly measure the amount of water that evaporated, and this over time. And then finally, in the more refined examples, we even had an environmental chamber with humidity and thermal control, where we could really have controlled lab experiments happening. So very complex, 5 years of work. And then we took this, we could prove the system in a two-dimensional setting, and we have the shape features noted. There's papers about it that you can access. And then we also tried to translate this effect in 3D. We worked with ceramic tiles. We made an experiment with 6 different shapes, with 3 different profiles in 2 different sizes, and reproduced those like facade-like tiles and tested them in the lab and also in an outdoor setting. And here that was very different because radiation of the sun changed everything. So this efficiency effect was totally overrun by wind situations and solar radiation. So that was also an interesting take. But we tried to translate those principles then together with a roof company into a roof tiling system. So we did a biomimicry design challenge with those findings. We did a workshop in Berlin with students that was called Breathing Skins. That also took on this topic of shape change with folding systems, and also introduced the leaf knowledge as an add-on. And so you see we had several really interesting results after only 1 week of workshop that we then also evaluated according to the main features that we were interested in. So that's another methodological approach, that we generate a lot of solutions and then do a proper evaluation according to the goals that we were interested in. That's not usually what you do in architectural design, or not so stringent with this research framework. But it's something that is really interesting if you want to push innovation. And I show this to you to, show you a glimpse of qualitative evaluation that you can do in your research projects or design projects too. And then also have another way of showing this in a visual. You see the best of projects immediately as an outcome. And then this was also translated into a research project that was carried out by my dear colleague Bernhard Sommer, who is here now. Hi Bernhard. And so I was moving back from the US due to COVID. So COVID upended a lot of the research that we did there. Also for the next project it was very impactful. And for this project we could take on the finalized PhD of Ariana and submitted funding for the Austrian Research Promotion Agency, and we got a 1-year study financed to do the project BioCool. And I had to sadly hand it over to Bernhard because I left to work with the funding agency, so I was, due to compliance issues, not allowed to take the project further. And I'm very curious about the report that is oncoming and to also learn about the result of this study. And so I want to switch over to the last project that we did in Akron, and this was about root growth. Another project really rooted in biology, and the project was informed by, again, by the context that I was in. So in Ohio, Ohio is at the Lake Erie. The Lake Erie has a lot of problems, among them also coastal erosion. The US in general spends billions of dollars each year for coastal stabilization and hurricane damage protection, and also to remediate damages. So foundation is a big issue, and coastal restoration is a big issue. And this is where this interest in root systems comes from. And together with two students from the integrated biosciences, we went about basic study about root systems in trees and how they could inform better foundation systems and improve coastal resilience. One of the studies is finished, so the foundation PhD is done. The other one is still ongoing. COVID has hampered our research for about 2 years, so we're still processing the results that we have gathered. So what we did is we again started with basic science, learn about the growth and morphology of root systems. We do our own experiments, so we scan and generate 3D models that we then analyze, gather principles, and then hopefully translate this into technical systems in the follow-up studies. So this workflow is really a very classical biomimetic one, but also, as you can see, it contains a lot reverse biomimetics too. So a classical root system looks like this. So this is a tree at a river. You see the root system is washed out. And also here multifunctionality, so trees have structural support by the root wad, and they are taking up nutrients, they are generating habitats, they are stabilizing the soil, and so on. So root systems, again, very multifunctional, and we are trying to research them. This is really difficult because roots are in the soil, so this was done manually for the longest time. So biologists were digging out roots, measuring roots, drawing the roots, so this was really done manually for the longest time. And what we are doing is we're scanning those systems. This comes with drawbacks. The biology is dead, so we have to kill the tree to take it out, and to use photogrammetry to really scan the coarse root system. This is another limitation. We have to remove all the fine roots because this photogrammetry and 3D modeling doesn't work with so fine root systems. So there's a lot of work involved before we can even start to do the scanning. For those of you who have never heard about photogrammetry, it's basically an image processing technology where you take pictures around an object, usually used for buildings, and then use a geometrical algorithm to generate a 3D object. And in the meantime, we have several really good softwares that can do this. So that's what we did with the root wads. So we generated 3D models that have their own complications, so it's a very complex thing. And on the way of this research, we refined this methodology. So we colored, we powdered, we kind of had, connectivities with markers, and so we experimented with a lot of different technologies to refine this process. We could have used laser scanning. Laser scanning is way less accessible, and it comes with other drawbacks. We also wanted to have a citizen science method. There will be a paper coming out about that too, so we also used a variety of different cameras for this research that we compared and tested. So there was a huge depth to this to this piece of research too. Finally, we had 3D models of those systems, and then we were going about to extract these morphological traits. So we wanted to understand the topology of the system, which is the kind of building principles that are abstracted. So how many parent roots, how many connectivities do you have, how many branchings do you have. So this whole topological aspect is really important. And then we started to also skeletonize the 3D model to have further output for biomimicry and biology. This was a really complex venture too, so we used Rhino and Grasshopper to go about this process. So you see here the different steps that it entailed, and here the respective elements. So we kind of cut the system in a certain array, then we defined the centers, we connected the centers, we connected the center lines, had a fault correctivity and so on. So if you want to know about the detailed process, please go for the publication. Everything is open access. And so finally we ended up with a skeletonized model that gave us even more information. And so you see here the topology of a detail, the center lines, the vectors of the root links, and so on. So we had a lot of information about those tree trunks that biologists usually don't have. So suddenly, with the tools of architecture and design, we can inform the biologists about crucial questions that they always had and never could answer. And this is what we call reverse biomimetics too. So suddenly we could answer those questions about the parent root and offspring root relationships, and we could answer questions that are important about our translation. For example, the porosity of the system. If we wanted to translate this into a foundation system, porosity would be one of the things that we could look at as a design feature. And this is something that we could then take over as a measurement and design technical foundations with similar values that would look entirely differently, because we could never produce such a complex 3D system. But we could take over some of those values and hopefully by this mimicking also the functionality. And another interesting piece was the angle between those root systems. So how does this angle change over this kind of root-wad system? So a lot of information that is abstractable and then usable as a biomimetic principle that we then take into a prototype system and then evaluate the prototype system according to the technological requirements. So the next step would be to prototype those artificial systems and then test them with wave channels and other like physical testing. Usually things are too complex to just calculate them. There's no simulation that tells you about this really yet. So there's also some criticism and some limitations about this process. So for example, we only had the coarse root system. There were limitations in occlusion, so it's a very complex 3D shape, so you don't see some of the features not on the pictures and so on. So there's also a lot of pros and cons that you can state. And again, here the publications that talk about this project. And for the translation piece, we have worked on this analogy between the biology and engineering very hard. There's a review paper that encompasses an analogy table, also with conceptual sketches of foundation systems that we could actually work on, that are simple enough to be translated. And one of the examples is this one. So this is one of the ways that we could translate this in an engineering system and use this knowledge that we have gathered from biology. So it's really a step-by-step introduction process, so we are not at growth yet. But I can tell you that colleagues of mine work on growing robots, and there will be other technologies out there that we can grow closer to these kind of growth principles too. So for the moment we are still in this kind of biomimetic stage, but I hope in the future this will change too. So the future of building foundations could look like this, that we have multifunctionality, the buildings can take up nutrients, can have like a water cycle system. You could use the foundations also to get rid of stuff, so it could be a transport system. It could stabilize the soil and the environment. The different foundation systems of buildings could communicate with each other and provide infrastructure on an urban scale and so on. And lastly, these systems could also be just coastal resilience systems that are introduced. So this is just a few projects that I presented here in kind of bigger depth or more depth as usual to illustrate also these time frames that are associated with the pictures that you see here. And lastly, I want to present some of the publications that we do. So I always strive for open access. It's not always possible to do the restrictions of the publishers, and in this case you have to contact me on ResearchGate and then I can send you a PDF in person, but we are not allowed to put it on the web somewhere. So if you're interested in one of the projects, just let me know. And some of the publications are also here in the front office, so the research projects that we did here in the Angewandte. You can also get the publications here, and I'm always happy to get questions, comments, and I'm always interested to collaborate. So you see a range of disciplines and a range of institutions that I have collaborated with in the past. So feel free to approach me if you want to do a project, or if we should go for some specific design or research idea that you have, and I'm happy to support or even be on board. And lastly, I want to stress the teams that I've collaborated with and the institutions. Biomimetics is not something that you can do on your own. You always need a lot of people to collaborate with. You always need a lot of different expertises from different institutions to play together. And so teamwork is essential, and it really takes also a lot of time to navigate a project. It needs time to understand each other and patience. So it's not a straightforward thing. And I'm also happy to advise you if you want to go about such a project too. And with this, I want to thank my collaborators that I cannot name in person in this framework. And I want to thank you for your interest and attention. I'm happy to take questions. Thank you.